Okay, so we are going to start our process of translation by taking the messenger RNA that we created earlier, we transcribed earlier from our DNA. Um, we're going to look at this as a genetic sentence. And I'm going to show you how you can take a genetic sentence and divide it up into genetic code words. And then we'll use those genetic code words to translate our message into protein. So again, I'm just taking our messenger RNA that we transcribed from our DNA earlier. So I'm just going to copy this over. So AUG and UGA. So a very short message it is. So now I'm going to show you how to be thinking about these genetic sentences. When you're looking at these genetic sentences, we're going to look at them in terms of three nitrogen bases, three consecutive nitrogen bases. So this AUG and then UGA will be two separate genetic words. So I am going to just separate them and just kind of start thinking about separating or looking at these genetic sentences as two separate pieces. And what you're going to call these two separate pieces, so we've got some terminology here. We call these codons, Codons. So a codon is a series of three consecutive nitrogen bases, or I could say a set of three consecutive nucleotides. Now, just to put a little bit of information in our notes here, a codon is a genetic code word, so fancy term for it, a codon. And these genetic code words, what they code for one of 20 different amino acids. So, amino acids, if you remember, they are the monomers or the building blocks of proteins. So, again, there are 20 amino acids, which makes proteins the most diverse group of biomolecules. If we were to take the RNA alphabet, AUCG, and make three-letter combinations, what we would end up getting if we took them and divided them into threes would be 64 different genetic code words, which is a lot of code words. So what that means is that some of your amino acids have more than one different code word. And I will show you how that you'll be able to tell the difference between them. Now, there's no need to memorize amino acids. We're going to look at the process of translating the amino acids using an external tool, which I just call a genetic code chart. So I have two different versions of this genetic code chart, and you'll also have access to them. This one is very similar to the ones that I've seen as practice on the SOL. It looks more like a wheel than a chart, but we're going to call it a chart. Now, the way that this works, and you see this little key, is that we're going to start on the inside and work our way out. So if you notice, the inside is divided up into four quadrants, and each of the four quadrants represents a base of RNAs. And then there are two other levels of letters that represent the three nitrogen bases in a sequence. So first, second, and third, and then outside is not a home run. Instead, it's one of the 20 different amino acids. And they're also abbreviated in sets of three. So sets of threes is a big theme here. Now, what we're going to do is we're going to do a little bit of practice with this wheel and then with a more traditional chart. So I'm going to start with the AUG. So first base, second base, third base, and then you'll find your amino acid. So start in the middle. There's your A. Then our second base is U. And our third base is a G. And if we look outward, there's our answer, which is methionine, which is represented just by MET. Now we're also going to try this with the traditional chart. So this traditional chart, works the same way. We're using our 3 bases from our codon to find our amino acid. So if we look on the left, we will see the first base. At the top is the second, and over to the right is the third. And where they converge is going to be your amino acid. So we're going to go through these AUG and find our answer. So the A, there's our first one, and it tells us, well, there's the U and the G, and where they all come together is going to be your AUG. So A is this row, then up at the top, our second letter U. So now we know the column. So now we focused in there on that little square. And then our G is going to tell us which row to go to in that little square. So take G, and there it is, same result, AUG, which is methionine. Now, this chart also has a key. They don't always have keys. And it lists this little section as the initiation codon, but there's a much easier way to say this. Simply stated, instead of initiation codon, we call it the start codon. Now, what the start codon means, it means that it is the beginning of the genetic sentence. It's the beginning of the gene. So it's saying translation begins here. Now, we're going to do this one more time, and we're going to be using the same process using the wheel and the traditional chart. All right, I do have a memory device here to remember that AUG is the start codon, and it has a little something to do with school. So when does school start every year? It might be the most dreaded month of the whole entire year, unless it's your birthday month. School starts in August, AUG. So it's a really easy, cheesy way to remember it. All right, so our last practice coming up here with the UGA. So go back to our wheel. So U, second base, G, U first, U, second is G, third is A, and then what we have here, we don't have an amino acid. Instead, we have what's called the stop codon, one of 3 stop codons. And it just means that this is the end of your gene. Stop translating here. Now we're going to see this same thing on our traditional chart. Here it comes. So first base is U, so there's our row. Second base is G, so there's our little block. And the third is A, and there's our last row. So UGA is the termination sequence, the termination codon, and it just means stop translating here. It's the period at the end of this genetic sentence. Now you can see there are 2 other stop codons, and they will be listed just like this. So unlike the AUG and the start codon, which you kind of sort of just have to memorize, you don't have to do that with the stop codons. So again, let's add this to our notes really quickly. So UGA is the stop codon. There are 3 of them. They will be listed. You don't have to memorize them. They are the ends of the gene. So whenever you reach one of those, that's when you stop the translation process. So you begin wherever you see AUG and you end wherever you see one of the stops.